So welcome everyone. Today we have the pleasure to be with Maria do Carmo Pereira and Joana Loureiro. I'm going to say a brief summary about them. Maria do Carmo Pereira completed her PhD in chemical engineering in 1998 at the Faculty of Engineering, University of Porto in Portugal. She is an associate professor at the University of Porto and the group leader of supramolecular assemblies at the Laboratory for Process Engineering, Environment, Biotechnology and Energy, LEPAVE. The main research areas that she is working on are nanotechnology and interfacial phenomena, supramolecular interaction, including novel nanoengineering biomaterials for therapeutic applications, nanocarriers to target the blood-brain barrier, nanostructured electrochemical immunosensor for detection of neurodegenerative diseases, and environment. Dr. Pereira is also co-founder of a deep tech startup, BNanoTech, and a patent related with nanoparticles comprising encapsulated drugs. BNanoTech was founded on February last year. The company's technology originated from research conducted at the Laboratory for Process Engineering, Environment, Biotechnology and Energy at the Faculty of Engineering at the University of Porto. The company develops controlled release system for transporting drugs to the brain. BNanoTech target marketing includes pharmaceutical and biomedical industries interested in advanced therapies for neurodegenerative diseases, with potential for strategic partnership and commercialization at the national and international levels. On the other hand, Joana Loureiro is an assistant professor also at the Faculty of Engineering, University of Porto, and senior researcher at the Laboratory for Process Engineering, Environment, Biotechnology and Energy. Loureiro did her bachelor and master's in chemical engineering, followed by a bachelor and master in pharmaceutical science. She received her PhD degree in chemical and biological engineering from the University of Porto in 2013. Since 2010, Dr. Loureiro has been working in the field of drug delivery nanosystem for brain disease treatment. She has an expertise in protein misfolding and pathological biomarkers associated with Alzheimer's disease. Her main research areas of interest comprise nanotechnology for biomedical applications, effects of fluorinated systems and peptides on the aggregation of amyloid beta peptide, and conformational studies of proteins and peptides, self-organizing system, and polymer surface. And today, both of them are going to give a very interesting talk, and the title of the talk is Nanoparticles as Drug Delivery Vehicles to the Brain for the Treatment of Neurological Diseases. So when you would like to start, we are ready. Thank you.
Thank you. We will share again because this stops. So elas têm o mesmo título. Aqui será as notas. Agora aqui que deve estar, aí que está para. Deixa eu. Okay, good afternoon. Sorry for my delay. I was trying to enter in the application, but was not possible. Thank you for, it's a pleasure for us to be here to present, not in your faculty, but here in Porto to present our company. And we also like to thank for the kind presentation that you done. BNanoTech is, we can say that is our newborn because it's a spin-off company that was born last year, and we are from the academy, and the BNanoTech is a good example of the research work that is done in the academia, and it's able to go to the market. It's a big challenge to create the company. We need lots of time available to be dedicated to the BNanoTech. BNanoTech is focused on target nanocarriers for brain disease treatment. And to create a company, we need to have a problem, and or we need to identify a problem, and after we need to arrange a solution for that problem. The problem that we are trying to solve is related with neurological diseases. Sorry, something is... Sorry, we had a technical... Can you see?
Yes.
Sorry,
yes,
sorry.
Now we see the presentation.
Sorry, something is strange here. Okay. Okay, let me go. Okay. 1 in 3 people suffer from neurological disorders, example Alzheimer disease, and at some point in their lives, regardless the age, making them one of the most significant public health challenges. What we know is that 99% of drugs fail in the treatment of neurological diseases, and they fail because they are not able to cross the blood-brain barrier. That is a tight membrane that we have in the brain. Currently, there is no company that has a BBB drug targeting program using nanoparticles as carriers. Sorry, it's not working. Okay, I'm trying to... Okay. What is our solution? What we propose? We propose nanoparticles that efficiently cross the blood-brain barrier by delivering therapeutic agents or drugs through surface functionalized nanoparticles. That kind of nanoparticles needs to have some characteristics, and the characteristics are high affinity with the brain. They need to induce brain permeability. The size required is in the interval between 100 and 200 nanometers diameter. They need to be stable and, of course, biocompatible. The technology that we proposed or our nanoparticles are lipid-based and polymeric. Lipid-based, we are speaking about liposomes. Polymeric, we use PLGA. These nanoparticles are surface modified to increase affinity to the brain and induce brain permeability. The administration of the particles will be intravenous. These nanoparticles reach the brain within 2 hours, successfully crossing the BBB, delivering the therapeutic agent directly to the brain. The system demonstrates biocompatibility, that is fundamental, versatility to transport several drugs, and exceptional stability, remaining stable for up to 7 months. Our particles have dual functionality. It not only transports drugs across the BBB, but also actively contributes to delaying one of the mechanisms of the Alzheimer disease. They are also able to be used for repurposed drugs that failed in previous clinical trials and reduce the dose needed to reach optimal concentration by the interval between 80 and 90%. So, our nanotechnology is a breakthrough nanotherapy that transports drugs, crosses the BBB, and delaying one of the mechanisms of Alzheimer disease. Why we speak about dual functionality. Here, it's represented the brain-blood barrier. I'm trying to find the laser you are seeing here is the brain-blood barrier. And we have our nanoparticles, the simple system that is lipid-based nanoparticles and polymeric and PLGA nanoparticles. We functionalize these both systems with fluorine, and we produce these nanoparticles that enter in the BBB by transcellular lipophilic pathway. And we can also functionalize with the transferrin, and the one possible mechanism to enter in the blood-brain barrier is by the mechanism of receptor-mediated transcytosis.
Sorry, Maria, we don't see the presentation.
No?
No.
No. Just this slide?
No, we don't see anything.
From the beginning?
No, we have seen it until just now.
Now you can see it?
Yes, now we see the presentation. Thank you.
Perfect. Okay. I think it was the let me confirm, this is this, you saw this slide?
Yes, this one we
okay. Okay, perfect. I forgot to refer also that our technology is patent pending. This slide, here I'm explaining the mechanism of internalization of our particles. And particles inside inside the brain, they could also delay the amyloid aggregation that is one of the biomarkers of the Alzheimer's disease. I have here represented in a simple way the mechanism of aggregation of the peptide beta amyloid. The peptides aggregate in oligomers, protofibrils, and forms externally to the brain cells amyloid fibrils that are responsible for the characteristics of the disease. This is just one of the mechanisms, because in Alzheimer's disease we have other biomarkers, but we are focused on the biomarker A beta. Here, I presented nanoparticles are two images from TEM image. Here we present the polymeric dual functionalized particles, and here we present the liposomes with the same dual functionality. In these particles was used in the proof of concept studies. We used in vivo model, mice, and from these results or these experiments can confirm or prove that our liposomes and also our polymeric nanoparticles enhance the BBB permeability and target delivery efficiently. Of course, we also tested the liposomes and the PLGA alone, and the part of the internalization in the BBB is significantly less. To finish the part of our proof of concept studies, we also done some experiments with our nanoparticles with the amyloid, the beta amyloid. And we follow the kinetic of the aggregation of the beta amyloid. I will not explain in detail the results, sorry, because I have here the list of... okay. Don't worry. Okay, we can see that the presence of surface modified liposomes, I have only the results related to liposomes, they alter the aggregation kinetics of A beta, delaying the initial nucleation phase. Because here we have the tag time of the T lag, while significantly delaying the amyloid aggregation by 60%. That is really a significant inhibition of the aggregation of this peptide. I think now Joana will speak a little about the bureaucratic part of our company.
Hello. Now we are moving from the technology to see the headaches of our company. So first of all, we start to see which are our competitors. It's something that we always need to see. And in the biotech company, it's even more complicated because, as you know, to put one biotech product in the market, it's even more hard. So first of all, we went to see well, I guess it stops to share again. Let me share it again. Can you see? Can you see the presentation?
Yes.
Great. So we went and we saw the big fish, which are Roche, Biogen, Eli Lilly, and we saw that they are also developing vectors to transport drugs to the brain. And at a certain point, we get happy because they are using some of them also the transferrin, which is one of our surface modifications. But none of them use the nanotechnology. So they are using, for instance, to modify the drug with an antibody or with transferrin to target the transferrin receptors, but none of them could do what we do. It's the nanoparticle. Imagine that it's like a ball and you can put the drug inside without any type of we take that advantage when we are comparing our assets with our competitors. So, and as Maria do Carmo said, our technology is very versatile because we can encapsulate different types of drugs in our nanoparticles to transport them to the brain. So this is our big difference when we are comparing with our competitors. Moving on to the market analysis, we would like to start just targeting the Germany and France, and you can ask us why that decision if you are in Portugal. Well, because they are two countries with high number of patients, so they have a huge population, so they have also high number of patients with the Alzheimer's disease. And at the beginning, we also want just to target the people with the Alzheimer's disease, because as we said before, our nanoparticles have a dual functionality, because we can transport the drug, but we notice that that particles also inhibits one of the mechanisms of the Alzheimer's disease. So now we are targeting Germany and France, and our decision, as I told you, was the market size of that two countries, and also they have higher power of purchase, which is very important, because when we are putting a new technology into the market, they will be a little bit more expensive when we compare with the other solutions that are already into the market. But of course, we want to reach the remaining Europe and also worldwide, if possible, for all neurological disease market. Also, we had to see our business model. So we discussed about the different strategies that we could take, but we finally decided to have a strategy, a B2B. Of course, our technology, it's very expensive, because, you know, we are talking about biotech, in vivo animals, clinical trials, everything, it's very expensive. But after we get the product into the market, and we expect, if everything goes well, to put the market in 2031, our revenues will be very high. So it's a very expensive technology, but also the revenues will be very high. But as the other biotech startups, we have a huge debt valley. So this is the debt valley. It's where we are at this moment, because we have the technology, but we need a lot of money to go and pass to the commercialization of our product. So, as you can see here, we just bring to you our roadmap to you to have an idea. We start this project a couple of years ago, and we participate in different accelerator programs. And last year, we get a financial support to open our startup, and it was our break point when we decide just to go to a university lab where we are right now and move to a spin-off. So also after that we deposit our patent, and it's already submitted, and we get an approval in Portugal, but we are entering other countries. In 2026, which where we are right now, we are trying to engage with CDMOs, and we expect to start prepare the human clinical trials next year. And in next year, we expect also to license our technology. And why you are talking about why we think in licensing? And of course, we are in 2026. This is what we expect in 2027, and maybe we can have some difference. But imagine 2027, we want to join a big pharmaceutical company, because as I told you, we want to expect to start the clinical trials, and they are super expensive. So we want to join a big pharma. Right now, we already reached the TRL4, and now we are in the middle of the TRL4 to TRL5, because we are finalizing our preclinical trials, the ones before the humans experiments. Also, we did several presentations. We went to some demo days. We participate, as I told you, in acceleration programs to get this background until to get the decision to get the startup. I don't know if someone that is there are thinking to open a startup, and if you are working in biotech, think that is a very long journey. It's not like the other high-tech products that the people just put easily into the market, easily and fast. Here it's not the case. It's a long journey. So it's stop again to share Give me a sec. It's also the last slide for you to have the last message that we want to give to you. So our BNanoTech, our startup, what they are developing is a solution. We are not developing a drug. We are developing, let's talk, a vehicle to transport our medicines to our brain. And with that solution, what we can do, we can repropose some drugs that, when tested before in clinical trials for the treatment of neurological disease, in specific the Alzheimer's disease, but just fails because they were not able to cross our blood-brain barrier, this barrier that we have in the brain. But with this vehicle, we can try again to see if we can make that drugs reach our brain in a sufficient amount. And also, we can test new drugs and make them to reach and to the brain and cross, of course, the blood-brain barrier. I don't know. This is what we have to talk with you today. I don't know if you have any questions, so feel free to ask. Sorry, I forget just because it was in the beginning. Our team is founded by me and Maria. As we told before, we are pure academics because we had our life here at the university doing research. But at a certain point, we decided to jump in this adventure. But our background is chemical engineer, as Anna said in the beginning. And over the years, we went through the direction to develop nano drug delivery systems. And yes, right now is what I have to tell you. So if you have any questions, feel free to ask.
Thank you, Joana and Maria. So we open the floor for questions.
I saw one here.
Yes.
Based on why lipid or polymeric-based nanoparticles specially suited and deliver drug to the brain? It is because BBB has lipophilic nature, which make them more suitable to lipid-soluble compounds, or other reason?
You can.
Well, the main reason is our main technology. It's not in the particle by themselves. It's in the surface functionalization of nanoparticle. And if you are thinking, as I told you, we are a nanotech startup. So we need to avoid new different things to put into the market. And we take advantage because the liposomes by themselves are already FDA approved, and also the PLGA, the polymer one that we are using, it's also FDA approved. So we take advantage of this and we choose to use that two types of materials to produce our nanoparticles. I don't know if it's the question or if it was clear. I think that Vanessa is writing. I don't know if you want to add something.
The functionalization with antibodies helps the particles to enter in the BBB or in the brain. We choose also these two kind of nanoparticles because it already exists companies that produce this kind of nanoparticles. They have the industrial methodology for the production, and it's easier to contract these kind of companies to produce the nanoparticles.
I don't know if anyone has any questions.
More questions? So I have two questions. The first one, are you planning to move to cancer therapies with your system? Because one of the problems is to reach the brain, and you have already accomplished that. And the second one, I work also with nanoparticles, and we always have problems with the liver, because the liver catch all the nanoparticles that we inject. So have you quantified the level of your nanoparticles that are captured by the liver?
Not directly. We studied the toxicity of the nanoparticles in the organs. Not we didn't quantify the accumulation of the nanoparticles specifically in the liver.
Okay. And are you planning to move to cancer therapies?
We we have other research line that we are both involved with, and we also use not this system but similar systems for the glioblastoma.
Okay, thank you. So anyone wants to ask something? No, so thank you very much. It has been a very nice presentation, and it has been very interesting how to see how to create a new company. So thank you very much.
Thank you for the invitation.
Thank you.
Bye.